Welcome to the UC San Diego Shiley-Marcos Alzheimer's Disease Research Center Brain Blast Series. My name is James Brewer, and I'm the director of the UC San Diego Department of Neurosciences and the Shiley-Marcos Alzheimer's Disease Research Center. I'm a professor of neurology, a clinical neurologist, and a brain imaging researcher. I'm pleased to present to you our virtual Brain Blast series. Each of the talks included in the series have been created by Shiley-Marcos ADRC colleagues with expertise in brain aging research that focuses on prevention, detection, diagnosis, and treatment of Alzheimer's disease and related dementias. Each of the experts featured in the series studies a unique aspect of brain aging and has developed a brief overview of their current research to highlight the state-of-the-art work that's going on here at UCSD and our center. We hope you'll take advantage of this free resource to learn more about the advances in brain aging, the importance of research, and the multitude of opportunities to get involved and participate in research studies.
Hello, I'm Douglas Galasko, and I'm going to discuss some of the research opportunities related to frontotemporal lobar degeneration through the Shiley-Marcos ADRC. Let's talk a little about frontotemporal lobar degeneration, or FTLD. FTLD consists of a group of progressive neurodegenerative disorders. Overall, FTLD is less common than Alzheimer's disease. However, it may represent as much as 40% of people who have cognitive or behavioral disorders due to a neurodegenerative process with onset before the age of 65. And the name frontotemporal lobar degeneration means that the frontal and temporal lobes bear the brunt of the brain pathology. The specific symptoms that arise depend on which brain areas are involved. FTLD is an umbrella term, and it consists of a number of subgroups of disorders, as illustrated in this slide. There are ways to approach FTLD depending on the clinical picture, the pathology, and what we understand about the causes. So starting off clinically, FTLD is divided into three main categories. People who start off with changes in behavior, called behavioral variant FTD. People who have changes in language, called progressive aphasia, and there are different subtypes of language problems, which I'm not going to discuss in detail. And then there are people who have some unusual movement disorders. The major variations are called progressive supranuclear palsy, or PSP, and corticobasal syndrome, or CBS. The next breakdown is in brain pathology. In general, the behavioral and language problems are associated with a protein called TDP43 that accumulates in the brain. Sometimes they're associated with a protein called tau that can accumulate, and sometimes with a few other kinds of proteins. In the movement disorder presentations, mostly they are caused by accumulations of tau, and rarely they may be due to other problems. Sometimes Alzheimer's disease may mimic corticobasal syndrome, for example. We don't know completely what causes FTLD. However, in general, we think about genetic factors that can be strongly causal or weakly contribute to FTLD, and then all sorts of other things that we don't understand that we call non-genetic or sporadic. In general, the behavior and language forms have some genetic contribution and some are sporadic, and the movement disorder forms are mostly sporadic. If we think about the relationship between clinical features and brain anatomy, when people have changes and degeneration in the frontal lobe, the clinical picture is usually that of alterations in behavior and personality, and things like difficulty with initiation and problem solving. And these can be put together to support a diagnosis of behavioral variant FTD. When the neurodegeneration affects the temporal lobe, there are changes in language. For example, difficulty naming things, trouble expressing full sentences or words, trouble understanding what words mean, and these are lumped into a category called primary progressive aphasia, or PPA. Sometimes the two may overlap. Some people with behavioral variant FTD can have aphasia, and sometimes the reverse may happen. Sometimes they can be relatively distinct from one another. Here is an example of a brain CNAD autopsy from somebody who had behavioral variant FTD, showing that there is marked atrophy or shrinkage or damage that is strikingly localized to the frontal lobe. If one looks under the microscope, I mentioned there are several different kinds of pathology, and the earliest pathology that was described in frontotemporal lobar degeneration was tau pathology. The variant called Pick's disease was actually studied and described in 1892 by a psychiatrist and pathologist called Arnold Pick. And he saw a patient who had progressive aphasia, and as illustrated in the slide, a prominent staining with an antibody to tau, so prominent accumulation of tau in structures in nerve cells that are called Pick bodies. Tau can accumulate in different areas and forms different kinds of inclusions, and these may differ in behavioral variant FTD, primary progressive aphasia, PSP, and corticobasal syndrome, and can actually be used to classify these disorders. So that would be the spectrum of tau pathology. Regarding TDP43 pathology, again, this is a normal protein that shuttles between the nucleus and the cytoplasm of nerve cells. And in many forms of frontotemporal lobar degeneration, TDP43 forms aggregates either in the nucleus or in the cytoplasm, which is where most of the cell is found, or in branches or neurites of nerve cells. And the slide simply illustrates different staining patterns that can be seen at pathology, and again used to describe subtypes of TDP43 pathology. So there are many unknowns about frontotemporal lobar degeneration and many research questions. On the clinical side, we're interested in accurate diagnosis, early detection, trying to characterize the variety of symptoms, and trying to determine ways to track change over time. Biomarkers have really made an impact on Alzheimer's disease research, and likewise in research in FTLD. And so looking at brain imaging and testing a number of different things using cerebrospinal fluid and blood can inform us about processes going on in the brain. It's obviously important to figure out causes and mechanisms, and there's a host of ongoing genetic research as well as other research into risk factors. And finally, treatment is a critical component of research. And in general for FTLD, this includes trying to do something to improve or control symptoms, but most importantly, to understand the disease and to try to slow down progression or even prevent the emergence of symptoms. So we're involved in several different initiatives regarding FTLD at UCSD. And one of them is to carry out clinical research. This is done through the Alzheimer's Disease Research Center, through our longitudinal program, together with a national study that is called All FTD. And the objectives of the study are to characterize, study, and follow people with behavioral variant FTD and PPA. In summary, this consists of interviews of patients and their caregivers or informants or family members, doing cognitive testing, obtaining a brain MRI, obtaining blood for DNA and plasma, and when people are agreeable, obtaining cerebrospinal fluid. We also provide social support resources through the ADRC and also by partnership with groups such as Alzheimer's San Diego and the Association for FTD. And we also provide genetic counseling and help people to be able to go through genetic testing whenever this is appropriate. And I'll talk more about genetics in a moment. So 20 to 30% of people with FTLD have some kind of genetic cause that is responsible. And this table shows the abbreviations or names of some of the genes that have been implicated in FTLD. The most common two are genes called C9orf72 and GRN, or granulin or progranulin, as it is sometimes called. However, there are other genes that may show changes or mutations. 4 of the genes listed in the table, C9orf72, granulin, TDP43, and a gene called VCP, are associated with pathological changes in TDP43. Abnormalities in the tau gene itself may be associated with tau pathology. And then 2 other genes that have been identified are genes called FUS and CHMP2B, and these are associated with different kinds of brain pathologies. So how can genetics help us? And the answer is by knowing a little more about what the gene might do, we can try to target the abnormal gene or pathways or proteins that are made as a result of the genes. And this may help us to develop personalized therapeutics for different forms or types of frontotemporal lobar degeneration. So as one example, there's been a host of exciting research looking at the gene for progranulin, which is a genetic cause of frontotemporal lobar degeneration. In people who are carriers of mutations for the progranulin gene, the mutations result in not enough of the protein progranulin being made. And so a number of different research efforts are underway that have led to clinical trials to try to restore progranulin. And we are a site in a phase 3 trial sponsored by a company called Elector. As I mentioned, patients with granulin mutations have a reduction in levels of a secreted protein called progranulin, and progranulin is critical for signaling within the brain. If it is decreased, then over time, people develop symptoms related to behavioral variant FTD or progressive aphasia. In this clinical trial, we will be studying an antibody that acts by preventing the breakdown of progranulin. What this does is to try to then restore the progranulin that is being made and also therefore restore its function within the brain. So for this trial, we are looking for people with frontotemporal lobar degeneration who either may want to be tested to find out if they happen to be carriers of a granulin mutation, or perhaps they already know that they are carriers or are related to somebody who is a carrier. If so, they may contact us, and if appropriate, they will be screened. We can try to enroll them in the study. If people do want to participate and qualify for enrollment, then there will be monthly study visits in which the antibody is infused intravenously, and the trial will go on for a minimum of 18 months. So we are interested and able to recruit people for the all-FTD and the progranulin clinical trial from not only San Diego, but from throughout Southern California and potentially from out of state as well, because we do have some funding to potentially support travel. So this last slide lists some resources for more information about frontotemporal lobar degeneration and our research studies at UCSD. I've listed a website for the all-FTD study itself, as well as a link to a description of the Elector clinical trial. We also are providing a link for the Association for FTD, which is a national organization that promotes support and research in frontotemporal degeneration. And finally, if people are interested and want to contribute and perhaps to discuss enrollment in these studies, they can call the ADRC at the number listed, 858-822-4800, and ask for our study coordinator, Yvonne Arias. Thank you very much.
We hope you've enjoyed this presentation from our UC San Diego Shiley-Marcos Alzheimer's Disease Research Center Virtual Brain Blast Series. Please look for other presentations in this series and share them with your communities. We hope that you'll also consider participating in brain research studies. We always have new opportunities for participation and are actively seeking individuals 65 and older without memory disorders, as well as those with a diagnosis of mild cognitive impairment or Alzheimer's disease and related dementias, such as frontotemporal dementia, Lewy body dementia, and Parkinson's dementia, as well as others. We're also proud to have a bilingual, bicultural team who conduct studies visits in Spanish. Your participation can make a real difference for future generations. Please click on the description below to complete the very brief survey about the presentation you just viewed. And for those who are interested and who live in San Diego, we'll be happy to contact you. Simply provide your contact information on the secure survey. Thanks for watching, and we'll see you next time.